Thank you for the introduction. Thank you for allowing me to present our work. I'm Karin Willems. I'm from IMEC. I'm a researcher there. And today I want to kick off the right meeting, the right part of this conference, talking a bit about dynamic addressing architecture for multi-electrode arrays towards ultra-high density DNA synthesis. So before delving into the technicalities, okay, that's difficult button. A little bit about IMEC. So IMEC is a world-leading research institute where we focus mostly on semiconductor technology, design, and application. And our primary mission is actually trying to bridge the gap that exists between academia and industry. We are headquartered in Leuven, Belgium, so not too far from here. But we do have sites all over the world, both technical and non-technical. Our Leuven site is pretty big and ever growing. We're still building, planning to build new buildings in the near future. Currently, we have two clean rooms, a 200 millimeter clean room and a 300 millimeter clean room. Both are staffed 24/7, and yeah, we can run all sorts of semiconductor processes there. To support that, we also have several wet and dry characterization labs, including biolabs, the typical university-style lab that you would expect, which is where most of this work was performed. Okay, so back to data storage. I think it's quite clear that in order for data storage to become a reality, there's many orders of magnitude that need to be bridged, both in terms of cost and throughput in the right part of synthesis. We can debate a bit how many exact orders of magnitude that needs to be. So this graph, I think this point here is 10 million sites on a DNA electrochemical array. So I think some tips might be here now. It's a lot, right? So that's not a trivial thing to do. Luckily, we believe that both cost and throughput can be tackled at the same time through density scaling, which means putting more synthesis sites on a chip for the same surface area. Note, however, that as mentioned a few times already, total cost of ownership is probably a more relevant metric, but I think we can all agree that cost of writing needs to go down still. And then a very quick recap. So DNA synthesis, as mentioned a few times already, is a cyclical process. In this case, we also use phosphoramidite chemistry. The coupling step is where you add a new base to a growing strand, and the deblocking step is the one that allows you to actually parallelize the synthesis, because this allows you to locally remove a protective group, and once this protective group is removed, the next base that you add will be able to couple only there. There's many ways that you can achieve this, like in the previous presentation by Hamid. With light, you can also do this with electrochemically generated acid using microelectrodes, which is what we want to focus on. How is this done in practice? The chemistry itself is quite old, I think more than 20 years now, but it's still more or less the same. You use hydroquinone and benzoquinone in solution, and as you oxidize hydroquinone electrochemically near the electrode, you release two protons in some form within the acetonitrile buffer solution, and this greatly increases the acidity, and this acidity then removes the protective group locally from the growing strands. And this process is what you can see here on this fluorescence micrograph. It's a time lapse of a small microelectrode array where we have a pH-sensitive dye in solution, and as you activate each electrode individually, they light up locally, and then you can create nice patterns. Come on. Yeah, it's a tough button. Just use this. Okay, so in the past 5 years, there's been quite some movement in terms of scaling these microelectrode arrays. So I took two examples that are publicly available. So one is the Helix system from Andrew Hall, who has pixel size of about 1.8 by 2.2 square micrometer. The array itself that he has is not too big, so 400 by 400, but in principle the density is quite high. Then there's the chip from Twist, which has quite a big number of electrodes, 128 million. Pixel size is somewhat smaller than in Helix, so 1 by 2 square micrometer. So that's quite a high density already. However, all these electrodes, they need some kind of switch underneath them in order to turn them on and off, because otherwise how do you determine which pattern to write? And typically this is done using this kind of CMOS circuit. It's called a 6T SRAM circuit. Typically it's still coupled to some other circuitry in order to drive the actual electrochemistry, but this is the base circuit that is used. So SRAM, that's typically used in CPUs as a on-chip memory because it's fast, it remembers its own state. It's not very space efficient because it uses 6 transistors. There's also other types of memories around, like DRAM, which uses a 1T, 1C, one transistor, one capacitor type architecture, which, because it only contains one transistor, is far more space efficient. And this is also what you see if you would plot the characteristic pitch between, oh, so it does work sometimes. The characteristic pitch between cells, bit cells. If you compare SRAM to DRAM, SRAM lags by a factor of 20 in pitch, so that means a factor of 400 in area. In addition, SRAM doesn't really seem to scale anymore beyond 10 nanometer. So even though you use the most advanced process node, your cell is still going to be the same size. DRAM, on the other hand, doesn't have this issue. And if you want to reach a characteristic pitch of 100 nanometer, you only need to use a technology node of about 40 to 30 nanometer, which is also quite a bit cheaper. However, DRAM is dynamic. It doesn't really remember its own state very long, and especially if you want to use it to drive an electrochemical reaction. So how does that work in practice? In theory, is that you have a memory array, which is addressed using rows and columns. So in this case, the rows address the gates of the transistors, and the columns represent the driving of the bit lines. So that means that if a column is on and a row is on, that means the corresponding cell will be on. And we don't address all the rows together, we do it one after the other, and we change the bit line pattern corresponding to which cells need to be activated and which need to be off. As this process happens, come on. Yeah. As this process happens, the speed with which you refresh each cell matters. If you have a fairly small, low duty cycle, which means the ratio between the time that each cell is on versus off is very, very high, then you have some initial electrochemistry happening at the beginning, but then as the charge gets consumed by the electrochemistry, you start to fall below the rate that is actually needed to perform the electrochemistry, and you're no longer performing the deblocking. If you have a very high rate, on the other hand, very high duty cycle, you can kind of maintain the potential at a useful state, which means the reaction will keep going indefinitely, which is what you want. However, in order to make such an architecture viable, you need to be able to address as many roles in a single cycle as you can possibly can. So a target we have in our minds is about 1,000. And which means you actually want to achieve the lowest duty cycle that you can get. And there are several things that you can play with in order to achieve that. One is voltage, and obviously the on time, the amount of time you take to charge up your cell. So we test this not on CMOS, but using a passive electrode array. So this is a small test device that contains an array of 36 electrodes in a ring disk pair configuration. So each disk at the bottom here is about 3 microns in size, and it's individually addressable using an external multiplexer. The counter electrode, which is in the form of a ring that sits 200 nanometer above that, is shared between all the other electrodes. And so if you fabricate that, this is what it looks like. This is just done with regular lithography, nothing too special. So using the same fluorescence, pH-sensitive fluorescent imaging, we set out to try and characterize this device when you operate it in a pulsing mode. So in this case, what you see is the same on time, so 50 microseconds. This is a limitation due to our tool. So in principle, the devices can, and I'll come to that later, can go much lower than that. But that's just the smallest on time that we could get. And we vary the off time. And as you can see, for at least 1.8 volts, the static picture is here at the bottom. If you leave it off for too long, you get a little blip of a pulse, but then the fluorescence decays very quickly. If you leave it on for off for a millisecond, you actually get the same kind of signal as you would for a static cell. And then if you look in detail at the high temporal resolution imaging, you can actually see the up and down moving, the charging and discharging of the cell and the potential and the fluorescence change that goes with it. And for 1 to 20, it's really like a static line. Now, like I said, we can also play with the voltage. So in this case, we keep on times the same, 10 milliseconds and 10 milliseconds. Off times the same and on time the same, except we change the voltage. And you can see that actually, while for 1.8 volts, we barely got a decent signal, for 2.5 volts, this is actually static. And the same kind of intensity as you would see with a DC signal. If you look into more detail, however, you do see that you merely shift up the baseline of this wave. So if you compare 1.8 volts and 2.5 volts at the same duty cycle, you just shift up the average intensity, and so the average proton concentration that you have. Finally, you can also do this in parallel. So in this case, those previous results were just for a single cell. You can also address multiple cells at the same time. So in this case, we switch cell A on for a brief period, we switch it off, then we go to cell B, then cell C, and then we leave it off for the remainder of the duty cycle, and we repeat this. And you can see, you can actually activate them all together at the same time, and they have the same kind of signal output. We also did some very small synthesis experiment. As a setup, we have a KNH as a DNA synthesizer. We modified it a bit with a flow cell and some other things to try and make it work. And inside that flow cell, we can insert our electrode chip, and then we drive that chip using a custom-built multiplexer and potentiostat. And all of that is controlled together using a custom Python API and scripting interface. So this is what the tools more or less look like. And so in order to show that you can effectively deblock using pulsing, right? Fluorescence of pH imaging is nice, but if you cannot really deblock that, it doesn't really tell you much. So in this case, what we did was synthesize a poly-A everywhere on the chip using just regular chemical deblocking with trichloroacetic acid. And then in the final step, we selectively deblocked certain sites with different conditions, electrochemically, either static or pulsed, and we coupled then a fluorescein amidite molecule as a reporter. And so only those sites that were deblocked efficiently will you see some fluorescence. And that's effectively also what you see. So in the case of the negative control, you don't see anything. In the case of the static, you see a nice bright ring, which corresponds to the silicon nitride ring between the counter and the working electrode. And in the case of dynamic, depending on how long you pulse, also this data needs to be repeated still a bit, you get similar behavior. So it does seem to work, which is also what is suggested by the fluorescence experiments themselves. Now, the device that we made is a very far cry from ultra high density, right? So it's 40 micron pitch, that's not 100 nanometer. And in order to make sure that whatever we find, we can extrapolate that to an actual realistic device, we set out as well to build a compact model, which is basically a circuit model that captures the dynamic behavior of such a system. In this case, it includes both the electrochemical part, so the electrode interface and the electrolyte, but also all the parasitics of the chip itself. Because especially if you work with dynamic circuits, time-based circuits, this is crucial because every capacitance needs to be charged and discharged, so that needs to be taken into account. In this case, every electrode is modeled as a double layer capacitance in parallel with a fire direct current element and a Warburg element to model the mass transport. So this really allows you to more or less accurately depict the behavior of the electrochemistry in an electrical circuit. And we also, we use both literature values but also extensive experiments in bulk for ourselves to parameterize the whole model, which is quite important. So if you then use this model, you can actually look into detail at what the currents are that you get out of each cell, what the overall potential at the disk is, and what the proton concentration above the disk becomes. So the red part here is during the charging pulse, so in this case also 50 microseconds. You get a very large current at the beginning. This is basically the charging of our track capacitance. We have a very long track, so it takes a lot of current to charge that. Once that is charged, the overpotential will start to build upon the disk, and only once that overpotential reaches a certain threshold will you be able to generate protons. And those build up quite quickly. And as you can see here, actually after less than 1 microsecond here, we could in principle already switch off the cell. So that would give us a factor 50 in terms of duty cycle that we can gain. But yeah, we couldn't go lower with the tooling that we have. Then once the cell is switched off, so that's the blue zone, you actually see that the electrochemical current, which is the blue curve, is completely supplied by the discharging of the parasitics. So the parasitics in this case are quite important because they're the reservoir that allow you to run the electrochemical reaction for as long as you can. So that also means that it's quite important that you have some high capacitance element inside the final circuit. So we mapped out, using the simulation, the possible duty cycles that we could have versus on time and voltage. So on the left-hand side, that's our experimental device. And it actually, the data that comes out matches quite nicely with what we see. So these kind of conditions, 2.5 volt, 50 microseconds on, 10 milliseconds off, that works, right? So that matches quite nicely. If we then use this same simulator, but instead move to a very aggressively scaled device, and in this case, we had to also increase the electrical double layer capacitance simply by making the electrode area itself quite a bit larger than just a ring disk electrode at the bottom. We can actually also reach 1000 to 1 if we increase the voltage high enough. In this case, the on times and off times are also of a different order of magnitude, which is what you would expect if you make a smaller device. Everything has to be faster. Okay, so with this, I'd like to conclude. So we do think that in order to further scale electrochemical DNA synthesis, we need to transition from static to dynamic addressing architectures, because those cell sizes are simply much smaller. The experiments and simulations that we have done so far do show that dynamic addressing can work and can also effectively deblock. And that, yeah, our simulations seem to agree quite well in terms of timescales. But in order to further develop this, we need to move to something more relevant in size, ideally with CMOS integration. So if anyone has a lot of money, you're free to come to IMEC and spend it to develop this with us. So thank you again for the organizers for allowing me to present this work, and thank my co-authors, in particular Senna, who did all the simulation work, and there's also Catherine, who's here in the audience, and Arnaud for supporting the work internally. And then there's also lots of other people at IMEC that have contributed one way or another to this work over the years. And with this, I'm happy to answer any questions.
So thank you, Kerim. Any question, comments? Okay.
Thanks very much for the very nice talk. Can you comment about your views on reusability of these chips? It seems, or let's say, yeah, for let's say DNA synthesis using electrode arrays, that reusability might be an important issue. Is that something you think is true, or?
No, that's definitely true. My experience is that even, so we use platinum electrodes, also they will randomly decay over time. It's difficult to make them stable. Definitely something that needs to be addressed in the future. But it's not, yeah, exactly the focus here. But it's been a frustration as well for us. Reusing the same device is convenient. And if you can't, then yeah, these things are all handcrafted, so then yeah, you run out of devices quickly, and yeah, it's not nice.
Thank you. Other questions, comments? Okay, one there. Two questions there.
Interesting presentation, thank you. Do you see extending it to lower voltages? Because as you go to smaller node sizes, the design rules won't let you get to even 1.8 volts, let alone 2.5. They're going to force you down below a volt if you're trying to get down to 100 nanometer electrodes and using a 13 nanometer node or something.
Yeah, this is not a trivial question to answer. I'm also not an ASIC person, I'm a biochemist. What I gather is that anyway, for this, it's likely you will need a non-standard process. Of course, if you need to integrate it as well, because part that I didn't show here is that you actually need a separate memory array still to store your frame of data that you're pushing to the synthesis array. Yeah, you need to be able to build that in the same logic, or you need to use some kind of multi-chip approach. Likely you will need a somewhat custom process in order to make this. Yeah, but I think the same is true for SRAM to a certain extent. You will also reach voltage problems as you scale down to the nodes.
Okay, one last question. First, there were a question there.
Is it possible to predict how many synthesis steps you'll be able to achieve on the, if you push it all the way to the smallest length scale that you presented?
With synthesis steps, you mean oligo length, or?
Yes.
In principle, yes. You're not talking about device durability, right?
No, just how long can you build?
Yeah, that's tricky to answer. It depends exactly on where you synthesize the DNA, and at this scale, crowding is going to be indeed an issue. I think CPG beads 50 nanometer pore size can give you like 100 mer at most before crowding becomes an issue. So it's probably going to be on that order. Yeah.
Okay.
Thank you very much, Karim.